Welcome to my channel Adhyan with Flow. Very warmly welcome, guys. Now, today's topic is polysaccharide. Wait, guys. Before starting with polysaccharide, I have to clear this term. Briefly, guys. Very briefly. I'm not going in a detail. Okay. Carbohydrate means simple. Only simple. Hydrates of carbon. That's it. Carbon, hydrogen, oxygen. That's it, guys. Now, if briefly we are going to discuss that we take carbohydrate into macromolecules, guys. Yes, means they are insoluble kind of nature. Okay, now carbohydrate divided according to their functional group, two types. Okay, aldehyde and ketone, and another one is alcoholic group. This is the symbol of aldehyde and this is the symbol of ketone and this is symbol of OH. You know, guys, what kind of amino acid contain OH, serine and threonine? Remember, you have to remember, guys. Okay, let's move further. Now, let's start with polysaccharides. Polysaccharides are long chain of sugar. Yes, guys. So, polysaccharides, they have poly means many, saccharides means sugar. Okay. Now, polysaccharides further, this is not a photosynthesis, further divided into homo polysaccharides. Okay. Another one is hetero polysaccharides. Okay, what is the special feature of homosaccharides? They have same type of sugar group and they have different type of sugar group. Like example is cellulose and their example is like pectin. Clear na, guys? Very clear. Okay, let's here. They are thread. Okay, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharides consisting of one type of monosaccharide. Look at here, guys. One type of. Cellulose is a homopolymer. Cellulose is a hemipolymer. Okay, same kind of sugar groups are present. Now, starch is also one of the example of, I'm writing here, starch. This is also a homopolysaccharide. Guys, starch divided into two parts, amylase and another one is, I'm taking you beyond your NCERT, amylase and amylopectin. What is the difference between amylase, amylose and amylopectin? They are made up with at least 500 to 1000 glucose units. They are present in straight chain. We can call it as a linear chain. Clear, guys? Linear. I show you. Okay, this is the linear, guys. This is the linear structure. They join in a linear form. Guys. But this is not a starch. Okay, this is not. So starch is a linear structure. They are not branched. Okay, clear or not, guys? Now, what is amylopectin? They are made up with beyond. They contain beyond 2000 to lakh, guys. Made up with glucose subunit, and they are branched. Compulsory, they are branched. Guys. Like this orientation. They are branched, huh? Bonding. 1, 2, 3, 4, 5, 6, and they connected with carbon number 1. Clear or not, guys? Clear or not? So starch is clear. I hope starch is clear. Now starch is clear. Okay. Starch is a variant of this but present as a storehouse of energy in a plant. Guys, polysaccharide, they work as a storage. There is one of the most important function is their storage. Storage, what they store? They reserve food, guys. I'm writing here, guys. They reserve food. Where starch is present? In plants, guys. I told you na, assume that plant make food with the help of photosynthesis. Plant, if they want to transport their food, the form is sucrose. This is also saccharide. Storage form is starch, guys. Yes or not? So where starch is formed? Chlorophyll. And where they store? Leucoplast. Storage. Everything can store. Protein can store, carbohydrate can store, everything can store in plants. Leucoplast. Okay, guys, let's move further. Towards the glycogen. This is the structure of glycogen, guys. Glycogen. Look at here, guys. Look at here. This is the structure of glycogen. Whole. Completely. Still they are branched. Glycogen is branched. Made up with alpha glucose. Repeating unit. All things are made up with glucose in plant, guys. Repeating unit, guys. Amount beyond 30,000, guys. 30,000. Okay, now let's move further. Okay, glycogen complete. Okay, inulin. Okay, guys, this is also important in kidney test. I know you don't know. Inulin is a polymer of fructose. Yes, guys, this is a polymer of fructose. What is inulin? I'm writing here. Inulin, they are made up with what? They are made up with fructose. Okay, writing. Oh God, my writing is fructose units. How much? Approx 30. Okay, special thing is no branching. What is the special feature of inulin? Okay, I'm written here insulin. It's inulin. What is the special feature? They preserve food in some plant species, and also they are study for kidney test, that kidney filtration properly doing or not. Okay, because our body can't digest this thing. Our human, this is the storage of plant. Okay, this is the storage of plant, but in our body we can't digest this thing. Okay, now let's move further. In a polysaccharide chain, the right end is called reducing end. Okay, and the left end is called non-reducing end. What they want to say that? Assume this is the single structure, monosaccharide. Assume if this strand is free, okay, if they don't pair with anyone, they are reducing. And if they are paired, look at here, they are bonding, they are free, carbon 1 is bind with another chain, they are non-reducing. Maximum are reducing sugars except sucrose, they are non-reducing. Plant transport their food according to sucrose form. Okay, next. Starch forms helical secondary structure. In fact, starch can hold I2 molecules in helical portion in starch. I2, okay, cellulose does not contain complex helical. Okay, note important. Okay, plant cell wall are made up with cellulose, and you know na what is cellular? Homopolysaccharide. Except these, chitin is also, I'm writing here example. Chitin also what? They are also homopolysaccharides. Chitin is also homopolysaccharide, and they made up with NAG subunit. And chitin, where they present? Chitin shell, chitin covering present in insect. Animal kingdom, phylum is insect. Okay, their shell, their outer covering is made up with chitin, guys. And heteropolysaccharide example is what? Pectin. Let's discuss pectin here. I already told you. Pectin. What is pectin? They are heteropolysaccharides. Clear? Very clear, guys. Now, what they do? They make cell wall, middle lamella of cell wall. Clear, guys? I hope now this is clear. And they made up with like everyone is made up with glucose, NAG. They are made up with NAG, and they are made up with some sugar, arabinose, not arachidinose, galactose, or galacturonic acid. This is very important, guys. According to your competitive exam, this thing is very important. I can't tell you guys how much they important. Look at here. The question is not arranged in a simple form. They rotate you like exoskeleton of arthropod made up with which kind of carbohydrates subunits? You have to tell nag. These are subunit and they made up with chitin, and this is the part of homopolysaccharide. Guys, one of the important. I also use this agar. Yes, I also use this. Okay, agar is a polysaccharide. Okay, this thing is clear. Agar is polysaccharide. You know, we can separate. We use agar in DNA fingerprinting, guys. Okay, in blotting technique, this is important. Seriously, I also done this thing with the presence of agar. Blotting DNA. How to isolate DNA isolation. When we give some heat, they form a jelly-like structure, and we pour DNA into them, and then this gel is taken in the apparatus of gel electrophoresis, and then DNA they run according to their molecular weight. If they are small, they run fast. If they are heavy, then run easy. So that's all about, guys. That's all about polysaccharide. That's enough, guys. That is enough. Okay, and now guys, next, nucleic acid. I already discussed in detail that what is nucleic acid. Again, I'm repeating briefly. This is the cell, guys. Cell contain nucleus and nucleus contain nucleic acid. We call it as a DNA. They are double helical structure and also contain RNA. They also wait. Single helical structure, and they made up with both made up with phosphate group, pentose sugar group, and nitrogen base. Same phosphate group, sugar group, and nitrogen base. Nitrogen base like adenine, guanine, cytosine, thymine. Adenine, guanine, cytosine, thymine. In my previous lecture, I already discussed in a very vast way, guys. I already teach you. So again and again teaching this thing, this waste of time. Okay, I already discussed. Look at here. Their structure of nucleotides contain phosphate group, sugar group, and nitrogen base. Nucleoside means it does not contain. They remove phosphate group, and remaining part we call it as a nucleoside. This is the structure of adenine. This is the structure of uracil. Okay, nitrogen base divided into two parts: purine and pyrimidine. A and G are purine, and C, T, U. What is they are? They are pyrimidine. I already elaborate this thing, guys. I already teach you everything. So repeating one thing again and again in the same chapter, I think waste of time. Okay. On our next video, we were going to discuss about, guys, little bit detail structure of protein. I'm gonna teach you how primary structure will form, how secondary structure will form, tertiary and quaternary structure will form. Okay, guys. So relax, and nucleic acid, you can rely to my previous video. Okay, again look at here. They also told figure 9.1. They already explain nucleic acid again and again. This is nonsense. Okay, this is a macromolecule. They are insoluble. When I say insoluble, then what do you think? They can't cross EM. So that's all for now, guys. Thank you. Thanks a lot, and please subscribe my channel, guys. Support my channel Adhyan with Flow. Thank you.